This is a peptide chain, made up of many of these amino acids. Each amino acid has a carboxyl end and an amino end. Every peptide chain also has a carboxyl end and an amino end that corresponds to the amino acids. This peptide chain is being digested, leaving the stomach and heading to the small intestine, where most of the breakdown takes place. At this point, the pancreas sends 5 enzymes to the small intestine to break down the peptide chain. These 5 enzymes arrive in helplessly inactive states: trypsinogen, proelastase, chymotrypsinogen, procarboxypeptidase A, and procarboxypeptidase B. Once in the small intestine, an enzyme produced in the duodenum called enterokinase activates trypsinogen into trypsin. With its newfound power, trypsin is able to activate more trypsinogen into trypsin, proelastase into elastase, chymotrypsinogen into chymotrypsin, and procarboxypeptidase A and B into carboxypeptidase A and carboxypeptidase B. Now, with all 5 pancreatic enzymes active, the real work can be done. All of these pancreatic enzymes arrive in the small intestine at once. As soon as they are activated, they start breaking down whatever bonds they can. Each enzyme breaks specific bonds. Trypsin, for example, breaks the bonds on the carboxyl side of the amino acids arginine and lysine, creating peptide chains with arginine and lysine on the carboxyl ends. Chymotrypsin breaks the bonds on the carboxyl side of the aromatic amino acids, tryptophan, phenylalanine, and tyrosine. This produces peptide chains ending in tryptophan, phenylalanine, and tyrosine. Elastase breaks the bonds on the carboxyl side of the aliphatic amino acids, valine, leucine, glycine, alanine, and isoleucine. Carboxypeptidase A acts on the products of elastase and chymotrypsin, meaning it acts on the same amino acids, tryptophan, phenylalanine, tyrosine, valine, leucine, glycine, alanine, and isoleucine. The difference is carboxypeptidase A breaks the bonds on the amino side of these amino acids, cutting off the ends to produce single amino acids and small peptide chains. Carboxypeptidase B acts on the products of trypsin, meaning it acts on arginine and lysine. Similar to carboxypeptidase A, it breaks the bond on the amino side, breaking off the ends to produce single amino acids and small peptide chains. In the midst of all this constant breaking down, the small intestine emits 2 more enzymes, aminopeptidase and dipeptidase, which further break down the resulting peptide chains. Aminopeptidase usually acts on peptides of 3 or 4 amino acids. It breaks off the amino end to produce a single amino acid, and may even do so once more on the same peptide chain. Dipeptidase acts on dipeptides, chains of 2 amino acids, and splits them into single amino acids. All of this happens simultaneously. Some enzymes will drift about until others have broken up peptide chains for them to act on. Eventually, the peptide chains are broken down into single amino acids and peptide chains of 2, 3, and occasionally 4 amino acids. These final products pass through the membrane into the cells of the small intestine wall. Inside these cells, the remaining peptide chains may be broken down further, and the cells can use the amino acids to build new proteins.